everyone. In this video recording, we're going to talk specifically about the auditory system, which includes hearing and equilibrium. So let's get started. Let's talk first about the hearing and then we'll talk about the balance. But the ear can be subdivided into three regions. You're going to have your external ear, your middle ear, and your inner ear. The external ear is going to be constituted of what we call the auricle, or your earlobe. It's going to be constituted of your external acoustic meatus, which you guys should remember from lab. Your tympanic membrane, which is this membrane that's made up of a very thin connective tissue, and it's very sensitive. And within the lining of the external acoustic meatus, you're going to have a gland that's called a ceruminous gland. This gland. And the ceruminous gland is going to produce what we call cerumen. Which is your earwax. Now this earwax is going to be important because it's going to help to protect the tympanic membrane, which, like I said, is a very sensitive membrane. And not only the cerumen protects the tympanic membrane, but also there are certain hair cells that are present here in your external acoustic meatus, which is also important for protection of your tympanic membrane. Now this is it for your external acoustic meatus. When we get to the not external acoustic meatus, your external ear. When we get to the middle ear, it's going to be this middle section right over here, and it's going to be constituted by what we call your auditory ossicles. So you have three auditory ossicles, which we can see on the next slide. Now, before we move on to the next slide, let me go over the inner ear, since I'm not sure I can go back to this slide during the recording. But the inner ear is going to be constituted of the oval window. It's going to be the first structure right over here. Then we're going to have your cochlea, which is the seashell structure right over here. We're going to have at the other end what we call the round window. And right over here you're going to have your vestibulocochlear nerve, which is your cranial nerve number 8, which is what's going to detect the sound, right? So we're going to have a part that's going to be connected to the vestibule and a part that's going to be connected to the cochlea. So that's why we call it the vestibulocochlear nerve. And before we move on, I want you to notice how your cranial nerve number 7, which is your facial nerve, is going to be very close to your vestibulocochlear nerve right over here. Okay, so basically these are the main structures. There is another structure right over here which you need to know, which is your auditory tube, and this is going to connect to your nasopharynx. So when kids, usually kids before the age of 2, they get a lot of middle ear infection, and that's because of this connection of your nasopharynx to your middle ear right over here. And so when they drink the milk, and usually they drink it laying down, and so the milk comes in here and sort of accumulates in the middle ear, and that's what causes middle ear infection. Another thing with regards to the external ear is that I told you that we have a lot of wax that accumulates over here, so cerumen accumulates over here, and we have a tendency of wanting to clean our ear up and use a Q-tip. And what happens is when you use a Q-tip, which is fairly big like this, you're just pushing the wax towards the tympanic membrane, and it accumulates right over here. So then your tympanic membrane can't vibrate during the hearing, and so it sort of interferes with your hearing. So they say that you shouldn't use anything smaller than an elbow to insert into your ear. Therefore, you guys should not use a Q-tip to clean your ear. The best thing to do is just let some water fall into your ear when you're showering, and then you can drain your ear out a little bit by tilting your head. But try not to use Q-tips. I know it's hard, everybody does it, but the best thing to do is that. Let's talk a little bit about how the sound is going to come into your ear so you can have hearing. And we'll talk more details later on, but you're going to have a sound wave that's going to come in here. It's going to bump into your tympanic membrane. Your tympanic membrane is going to vibrate, and it's going to make your auditory ossicles move. Now, we didn't talk about the auditory ossicles yet, but you do have 3 auditory ossicles. The first one is your malleus, right over here. So this is your first one. Maybe I should change color. So your first one is your malleus, your second one is going to be your incus, and your third one is going to be your stapes. Now if you notice over here, once your tympanic membrane vibrates, it causes your auditory ossicles to move. Once they move, the stapes is going to hit the oval window. Once it hits the oval window, it's going to circulate into your cochlea, and once it circulates into your cochlea, we're going to see later on that it's going to stimulate what we call the basilar membrane, which is a membrane that's located internally inside the cochlea. And then the sound will come out through the round window, and basically when it does this, it's going to stimulate your vestibulocochlear nerve. Okay, so we'll talk later on more about this, but I just wanted to make sure that we use this figure. On the next slide, we can a little bit better the middle ear ossicles. So here you're going to have your malleus, which is your first one. Then you're going to have your incus, which is your second one. And the last one is your stapes, which looks like a little bell. So if you think of Miss, then you won't forget the order of these middle ear ossicles, or your auditory ossicles. Now before we move on and talk about the inner ear structures, this is sort of a little graph that sort of summarizes what part of the inner ear is going to give you the function of hearing, and what part is going to give you the function of equilibrium. You guys will understand these structures a little bit better when we get to the next figure, but basically you're going to have your membranous labyrinth, which is going to contain your basilar membrane, which we're going to talk later on. This inner structure is going to be filled with a fluid that's called an endolymph, and it's going to be surrounded, the membranous labyrinth is going to be surrounded by what we call a perilymph. And on the next figure, we're going to be able to see it better. But your cochlear duct, which is the inside of your cochlea, is going to be responsible for hearing. Then we have this other structure that we call the vestibular complex, which is the one that's going to give you information and function of equilibrium. And within the vestibular complex, we have these ducts that we call the semicircular ducts, which you guys are going to see on the next slide. And also these two other structures that are called the utricle and the sacculae, which are going to be important for gravity and linear acceleration. So that we're going to see later on. So like I was saying on the previous slide, you can see right over here in the middle, you're going to have this structure in blue, which is what we call the membranous labyrinth. So it's a membranous or a membrane that's going to be filled with a liquid that's called endolymph. And then the brown part is what we call the bony labyrinth, which is going to be filled with perilymph. So it's a liquid on the outside. Now let's talk about the semicircular canals and ducts. So this is what's going to be part of this main structure that we call the vestibule. This is what's going to give us equilibrium. So we have 3 semicircular ducts, which the first one is going to be the anterior semicircular duct. Then we're going to have a posterior semicircular duct. Then we're going to have what we call a lateral semicircular duct. And if you notice, these ducts, they're all positioned differently, and this is what's going to give us the sensation of equilibrium. And we're going to see how this works. But before we talk of how this works, let's talk about the structures within the semicircular ducts. So right at the beginning of these semicircular ducts, we have these structures that are called ampullae. So it's just an enlargement of the ducts. And these ampullae are going to contain what we call cristae. And the cristae are going to be structures that are going to contain hair cells. Now, each of these hair cells are going to contain what we call a kinocilium and a stereocilia. And we're going to see that on the next slide. But before we move on to the next slide, I just want to show you guys a couple more structures. So I want you to remember where your maculae is, which is this structure right over here, and your utricle, which is this structure right over here. And we're going to get to it later on. And then we have one more structure, which is the sacculae, which is this structure right over here. Okay, so the sacculae, if you notice over here, the sacculae is going to contain the maculae. So just remember when we get to talk about these parts later on. Now let's look at this next slide, and we're going to be talking about the cristae within the ampullae, which is the initial part of the semicircular ducts. So if you notice over here, the semicircular ducts are going to contain the ampullae. Within the ampullae, we have the cupula, and the cupula, like I said, is going to contain hair cells. And if we look closely at these hair cells, you're going to notice that we have 2 different types of structures. We have what we call the kinocilium, and then we have the stereocilia. The kinocilium is going to be this large hair cell, which is the first, always the first hair cell. So it's the bigger one of them. So we call it kinocilium. I always think of kilo for kilogram, so means big. So that's what reminds me of kinocilium. So it's a big structure, a big cell. And the others are going to be called stereocilia. Now this is how we're going to be able to stimulate our nerve right over here. Okay, so pay attention how we're going to be able to stimulate this nerve. And if you notice over here, all these structures over here, you're going to have several of these structures together on this side, and they are going to form your sensory nerve. Okay, so several of these sensory nerve endings are going to form your sensory nerve. So let's see how this occurs. So basically, remember that this ampullae is going to be located at the beginning of every semicircular duct. So depending on how you move your head, because your semicircular ducts, they're going to be positioned in different angles, right? So you're going to have an anterior, a posterior, and a lateral semicircular duct. And depending on how you tilt your head, basically, if you tilt your head from right to left, what's going to happen to the stereocilia? Basically, they're going to be going on top of the kinocilium. And if you do this, it's sort of like, for me, when I think about this, I'm thinking about the stereocilia is going to be stimulating or tickling the kinocilium. So this means that we're going to stimulate the hair cell. If we stimulate the hair cell by stimulating the kinocilium through the stereocilia, then we're going to activate the sensory nerve ending, and we're going to be able to send the impulse. Now if the movement goes on the opposite direction, so if it's going in this direction, I think of always like the big cilia, which is the kinocilium, is going to crush the stereocilia, and by crushing the stereocilia, it's going to inhibit the signal. So if it inhibits the signal, then you sort of block the signal from going through. Okay? And by doing this and tilting your head to one side or to the other side, you're going to either be stimulating the anterior ampulla, or you're going to be on the opposite, inhibiting maybe the posterior and the lateral. Or if you maybe stimulate your posterior, then you're going to be inhibiting your anterior and lateral. And by doing this, either stimulating one and inhibiting the others, it's sort of going to give you your equilibrium, because it's going to give you where you are at that specific time. And this is what gives you your equilibrium. And here, this is just to show you exactly what's happening. So if you're tilting your head, depending on the angle that you're tilting, you can see that the anterior semicircular duct for yes, that means you're stimulating this part. And then you're saying, actually this is not doing that, but it's showing like which semicircular ducts are going to be moving when you do certain, or stimulated when you do certain movements of your head. So if you're moving your head in the yes direction, so up and down, you're going to be stimulating your anterior semicircular duct. If you're moving your head in the no position, so from one side to the other, you're going to be stimulating your lateral semicircular duct. And if you're tilting your head backwards, you're going to be stimulating your posterior semicircular duct. So these are just ways, and you can see where the semicircular ducts are going to be located. So right next to your internal acoustic meatus. I forgot to mention on the previous slide that when you're tilting your head to one side or to the other side, what's going to be moving the cupula is going to be the endolymph that's inside of your semicircular ducts. So it's either going to cause the kinocilium to bend on top of the stereocilia, and if this happens, then you're inhibiting the signal, or it's going to cause the stereocilia to bend on top of the kinocilium, and if that happens, then you're going to be stimulating the transmission of the signal. So now we're going to be talking about the utricle and the sacculae. The utricle and the sacculae, they're going to be connected to the ampulla, and they're also going to be connected to each other and to the fluid within the cochlea, right over here. So the hair cells that are present in these structures, the utricle and the sacculae, they're going to be present in clusters. So you can see these hair cells right over here. These clusters, they're going to be called maculae. Now these hair cells, the interesting thing about these hair cells is that they're actually going to be embedded in this gelatinous material right over here on top. That's going to be consisting on top of it, you're going to have these structures that are called statoconia. So right over here. So let's talk about this again. Okay. So the hair cells, which are called maculae, they are going to be or contain a gelatinous material on top of them. So gelatinous material on top of the hair cells, and on top of this gelatinous material, we're going to have these structures that are called statoconia. And statoconia are calcium carbonate crystals. And this gelatinous material, together with the statoconia, they receive the name of otolith. Okay, so when we put gelatinous material together with statoconia, we can call it otolith. And how are we going to be able to stimulate these hair cells? So basically, because the statoconia is made up of a sort of heavy material, so-called calcium carbonate crystals, when you tilt your head from one side to the other, you're going to be sort of moving the statoconia, which moves the gelatinous material, which then moves your hair cells. And by moving your hair cells, it's going to stimulate your nerve, which is the yellow structure. Okay, so you can see right over here on this structure on the bottom. So if your head is in neutral position, basically the statoconia is going to be on top and nothing is going to be moving. But if you tilt your head posteriorly, then the statoconia sort of moves, moving your gelatin and therefore moving the hair cells. And this, in return, stimulates the hair receptors. Now here we have the cochlea and the organ of Corti. Basically, I think it's easier for us to see on the next slide how this is going to happen. So you can see over here, this is how you're going to detect sound. So basically, this is part of your cochlea, right over here. Within the cochlea, you're going to have these structures here that are called organ of Corti. And if we amplify these structures, you're going to see that you're going to have an outer hair cell right over here, and then an inner hair cell right over here. Now this is how it's going to happen. So within the organ of Corti, you're going to have what we call a basilar membrane down here. And on top, we're going to have another membrane, which is called tectorial membrane. So what happens is, the same thing is going to happen in terms of moving the hair cells. But if your basilar membrane is moving in this direction, maybe I should put the arrow further down for the basilar membrane. So if your basilar membrane is going to be moving in this direction, this means that the tectorial membrane is going to be moving in this direction. So if the tectorial membrane is moving from right to left, and you can see how these cells are going to be, these hair cells are going to be larger, therefore they're the kinocilium. That means the stereocilia is going to be stimulating the kinocilium, and therefore you're going to have activation of these receptors. Now, if the basilar membrane is going to be moving in the opposite direction, if the basilar membrane is going to be moving in this direction, this means that the tectorial membrane is going to be moving in this direction. And if the tectorial membrane is moving from left to right, it means the kinocilium is going to be flattening the stereocilia, and therefore you have inhibition of the signal. So you always have to see where the movement is going to be. So if the stereocilia are moving towards the kinocilium, you have activation of the signal. If the kinocilium are flattening the stereocilium, then you have inhibition of the signal. So it's sort of the same basic type of mechanism, where if you're activating the kinocilium to move towards the stereocilia, then you have inhibition. If you're having the stereocilia activate the kinocilium and move on top of the kinocilium, you're going to have excitatory mechanism, which is going to send the signal of sound detection. So this is the pathway for our auditory sensation. So basically, if you have the stereocilia stimulating the kinocilium, it's going to travel through your vestibular cochlear nerve, which is going to go through all these steps, which we don't have to know. But at the end, it goes through your thalamus, and from your thalamus is going to go through specific areas of your brain for detection of sound. Now, we're going to see a recording or an animation in lecture that's going to show you how we're able to differentiate low frequency sounds from high frequency sounds.